All right. Hello, everyone, and welcome to today's CryoEM Current Practices webinar. You're joining the 3 national centers established by the NIH Common Fund Transformative High Resolution Cryo-Electron Microscopy Program. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for CryoEM Access and Training, NCCAT. With me here today is my colleague at NCCAT, Ed Eng. Lauren Vega is here from the Pacific Northwest CryoEM Center, PNCC, and Michael Schmidt is here from the Stanford SLAC CryoEM Center, S2C2. This webinar is a regular event that we host the last Thursday of every month. Along with the exciting biology we hear, all of the talks we host highlight the methods that research groups are using to obtain and interpret the cryo-EM data they can collect with the help of National Center resources. We hope these talks are useful, especially for groups just starting to use cryo-EM. We are still working out the final logistics for next month's speaker, but look out for announcements from our mailing lists, the 3D-EM and CCP-EM bulletin boards, and also the information will be available at the cryoemcenters.org website on the events page that you can see the address for there. Today's speaker, Dohoon Kwon, is joined today here by his mentor, Professor Suk-Young Lee, and Dohoon was invited by PNCC, and Lauren will introduce him properly after my colleagues from each of the centers give a short update on the cryo resources we make available at no cost to the research community. So a couple final logistics: we are recording today's talk, and you can find the recording along with those from our past talks on our YouTube page or through the events page at the cryoemcenters.org website. Please use the Q&A button to send questions at any time during the talk. You can also upvote questions you see there, and Lauren will be moderating the Q&A at the end of the talk. And so with that, I'll turn over for introductions from our centers. And Ed, you're up first.
Hello, I'm from NCCAT here at the New York Structural Biology Center. We offer high-end instrumentation access on one of our 4 dedicated Krios instruments. We offer cross-training through our embedded researcher program, as well as screening on our plunge freezers and Glacios instrument. We have a quarterly call for proposals. The next one is actually tomorrow. I would like to point out our sister center, Compa, is having their second symposium on memory production and analysis. That'll be June 22nd to 24th. We have a lot of confirmed speakers for that. For example, Wolfgang Baumeister, Brian Kobilka, Bob Stroud, Hao Wu. So if you want to find out more, you can contact us at the NCCAT user office. I'll turn it back over to you, Christina.
Thank you, Ed, and I'll turn right over to Mike.
Good morning, afternoon, evening, or whatever. I'm Mike Schmidt, and I'm one of the co-directors of the Stanford SLAC CryoEM Center, S2C2. We have 3 Krioses and a Thales Arctica for screening. We have a full program of high-resolution data collection, training on all aspects of cryo-EM, and are developing a screening application process where people can come either sending their grids prepared at home or having us and them prepare the samples here for screening. We're excited for all these programs, and please apply at cryoem.slac.stanford.edu S2C2. Thank you.
Thank you, Mike. And I'll turn over now to Lauren.
Hi, everyone. So my name's Lauren, and I'm the project coordinator for Pacific Northwest Center for CryoEM. So we have 2 proposal types now. We have a screening access that gives up to 5 days of on-site sample preparation optimization that we have targeted to low resource institutions. We also have our general access that has both single particle and tomography projects, which gives up to 480 hours a year and is valid for up to 2 years. Our general access proposals are eligible for all of our training workshops at PNCC, and we do have a monthly submission deadline for our applications. Each of our applications that are approved do get delegated what we call a scientific point of contact, or a SPOC, that you can use as a resource and do run your data collections on our microscopes. So for microscopes, we have 5 of them. We have an Arctica with K3, 4 Krioses with K3 and Falcon 3. 3 of our Krioses are equipped with energy filters, and 3 have also been upgraded to fringe-free imaging. And then for sample preparation, we have a VitraBot, a Leica GP2, and a VitraJet, which is unfortunately still undergoing validations before being offered to users. And then lastly, I want to mention that we're now able to finally have visitors on site. Our users can book to have in-person visits as of this month. And as of next month, we'll have our first on-site workshop. So we do have a microscope operation workshop that we've also announced that will be occurring in June. And we're also rolling out a new data collection workshop for our general access users sometime in September. So please keep an eye on that for those that are interested and apply today. I want to go ahead and introduce our speaker as well. So Dohoon Kwon is currently a postdoc for Suyok Young's Lee Lab at Duke University School of Medicine. He received his PhD from Korea University, where he performed structural studies of members of the autophage pathway, and he joined the Lee Lab after graduation in 2019. His current work focuses on studying temperature sensing trip ion channels using cryo-EM, which is ironic since he doesn't prefer spicy food. So with that, now I'll hand over to Dohoon, his talk, Heat-Dependent Opening of TRPV1 in the Presence of Capsaicin.
Okay, can you see my screen? Right? Okay, let's get started. Okay, thanks for kind introduction, Lauren. Hello, everyone. My name is Dohoon Kwon. I am a postdoc at Suyok Lee's lab at Duke University. It's a great honor to share my research result with you. Today, I'm going to talk about the heat-dependent opening of TRPV1 in presence of this capsaicin. We all know we have the 5 basic human sense: vision, hearing, smell, taste, and somatosense, or the somatosensation. There are the several types of the somatosensation, including the thermoreception, temperature sensing, and mechanoreception, pressure, vibration, and proprioception, and noxious, pain. The main topic of today's presentation is the thermoreception, temperature sensing. Many receptors are related to thermoreception. Among them, TRP channel is best known and well studied. The TRP channel is an ion channel superfamily consisting of 6 subfamilies and 28 members. Some TRP channels are known to detect the temperature, which we refer to as the thermoTRP channel. 8 thermoTRP channels has been reported until now. Each thermoTRP channel covers a different temperature range. For example, the TRPM8 is activated by 18 to 24 degrees Celsius, with for TRPM8 the pore sensor. And TRPV1 is activated by over 42 degrees Celsius, with for TRPV1 the heat sensor. Today, I will focus on TRPV1 in this presentation. TRPV1 is well known as a capsaicin receptor. David Julius group first identified the TRPV1 in the late 1990s. They found the TRPV1 is activated by the various stimuli, such as this vanilloid compound, capsaicin and resiniferatoxin, and proton, especially the low pH, and the noxious heat over 42 degrees Celsius. Along with the other thermoTRP channel, TRPM3, MA, A1, and M2, TRPV1 play a crucial role in detecting noxious heat at the sensory neuron. I think that many people might already know the TRPV1 cryo-EM structure was solved in 2013 by David Julius and Ethan Cheng's group. Now, their work was the groundbreaking in structural biology and the TRP channel created both. As far as I know, this TRPV1 structure was the first atomic resolution structure using the cryo-EM, the resolution revolution. As you can see here, TRPV1 is a homothetrameric structure like other TRP channels. And this is the architecture of the single subunit of TRPV1. This is the direction of this polypeptide chain. Here is the N-terminus, and it's going through the transmembrane domain and out to the cytoplasmic domain again. In the transmembrane domain, there are the 6 helical segments for S1, 2, 3, 4, 5, 6. Among these, the S6 helix is the gate helix, and the PH and the PL represented is the pore helix and pore loop. These pore helix, pore loop, and S6 gate, they form the channel spore. In cytoplasmic domain, consists of the anchoring lipid domain and coupling domain and trip helix and C-terminus domain. This is the trip helix, interfacial helix followed by the S6 gate. It's common feature among the trip channel. Yeah, just to summarize, these are kind of outstanding questions of TRPV1 we have. First thing is the mechanism of the heat-dependent gating of the TRPV1. Second thing is the location of the TRPV1, the heat sensing domain. This type of this ion channel, the heat sensing ion channel was not well studied in terms of the structural biology. So many people the question about the heat sensing domain. And the last one is about the temperature sensing model. Many groups they propose a heat sensing model of the thermotrip channel, but there is no structural evidence yet. So to prove this question related to heat dependent gating, we developed a high temperature bridging method. As you already know, the general plunge bridging method is performed at this relatively low temperature, 4 degrees, 8 degrees, sometimes it's the room temperature, right? But in the case of the TRPV1, you can imagine, right? The activation temperature is around 42. That means that it requires to heat up all equipment. So if someone wants to try this protocol, the high temperature bridging, I just want to emphasize the preheat all tool, which contact with the sample from the tip, tube, a tweezer, grid, and even the blotting paper as well. Basically, our bridging machine setting is the 48 degree, the 8% humidity. It might be possible there are some heat loss during the plunge bridging. So we set up the temperature a little bit higher than the threshold, the 48 degree. The threshold is around the 42 degree. In the case of the sample, we incubate the protein at the 48 degree for 30 seconds in the dry bath. After the incubation, they apply the sample and freeze the grid without delay. I will show you the actual micrograph and result. This is the example of this 4 degree sample. You can see in the low magnification image, the ice thickness looks normal in terms of the distribution. There is no any very thick area and very thin area, right? Also, the particle distribution is just fine. This is the high magnification image. You can see, you can find the individual particle, this one, and this is the side view, and this is top-down view easily, right? And this is the result of the 2D classification. This is the example of the 48 degree sample. You can see in this low magnification image, the ice thickness is very, very extreme. Yeah, here is the very thin, it's almost the empty side. Whenever we check this area, the ice is melting or it's just empty. Also, this area is a very thick area. So only this region, very narrow region, we can collect. Also, we can see the clear differences in the high magnification image. Here, many particles they form the aggregation like this, but here we can find some intact particles like this one and this one. And this is the result of this 2D classification. compared to the 4-degree the sample, 2D classification is also different. I will show you this next slide, this comparison. Yeah, this is the side-by-side comparison. You can see the 2D classification result. There is the clear differences between this side view and this side view. I will show you the detailed explanation later. Okay, so actually we already optimized this protocol, the heat high temperature freezing protocol quite a bit. We tested detergent sample as well, but not screen all of detergent, but tested the digitonin and the DMCHS. But the most particles are falling apart in that condition. Also, we tried the several different temperature setting and incubation time. Our current protocol is the nanodisc and 48-degree and 30-second incubation, and using the ultrafoil. And also we stick with the LICA. Using this method, we solve the several TRPV1 structure with various conditions, various temperature conditions. Okay. We solve the nanodisc structure in various temperature conditions using that method. 4-degree and 25-degree and 48-degree. I will explain all of this structure one by one. This is the map quality, and this the oil distribution and this local resolution information. Next, is the descent quality. Okay, let's get into the main. First, we solved the 4-degree APO structure nicely. You can see here for the control experiment. And then we solved the 48-degree APO structure like this one. We tried a lot, but we could not get an open state without any ligand. So we found out only the very N-terminus region was the less pleasurable. There is the density, but this quality is very bad, so we cannot build a model. Also, these differences, we can check the local resolution image. So yeah, this part is almost gone, right? And then even the 2D classification result, we can see the clear differences. Yeah, this part, we cannot see any of this very N-terminus region, right? Okay, this is the pore focused view of the 4-degree APO structure. And this is the overlay of the two models. The yellow one is the 48-degree APO structure. You can compare these two. These two is almost identical. We cannot find any conformational change between these two. And distance of the two gates is the identical. I missed this. Actually, the TRPV1 has the two gates. The one is the selectivity filter and one is the S6 gate. This both gates is the identical, so there's no any conformational change. This observation led us to reason that TRPV1 in nanodisc condition is more difficult to open than the cell membrane condition, the electrophysiology condition. The intrinsic gate equilibrium of the TRPV1 may be influenced by the physical and chemical differences between two different environments. For example, the lack of the membrane voltage across the nanodisc system, and different lipid composition and lateral pressure profile between the two systems. So we thought an additional stimulus was required to capture the heat-activated open state of TRPV1 in nanodisc condition. We searched a lot of paper and tested some ligands. Among them, we choose to utilize capsaicin for another stimulus because it is well known that heat and capsaicin activate the TRPV1 independently. Consistent with the earlier study, we found although this 100 nanomole the capsaicin, you can see here, 100 nanomole capsaicin does not elicit the activation current, right? It does sensitize the TRPV1 to heat activation with increased efficacy. We can compare this. This is the without capsaicin pre-treatment, and this is the with capsaicin pre-treatment. The efficacy is very different. Also, we measured the Q10. Q10, the temperature coefficient, is this measure of the temperature sensitivity based on the chemical reaction. We can calculate the Q10 using this equation. For most of the biological system, the Q10 value is the 2 to 3. The Q10 of the thermotube channels are over 10. As you can see here, this is the control and this is the pre-treatment of the capsaicin. The heat sensitivity is not substantially affected by pre-treatment with capsaicin. We already confirmed the heat itself could not activate the TRPV1 in this condition, right? And then we also want to check whether capsaicin can activate the TRPV1 in this condition. So we solve the structure at 4 degrees with the capsaicin. You can see this figure. The high quality 3D reconstruction allows us to assign the configuration of capsaicin in TRPV1 structure unambiguously. It fits very well. Compare with this 4 degree APO. The 4 degree capsaicin-bound structure has shown a slight conformational change around this vanilloid pocket due to the capsaicin binding. This red one is the capsaicin and blue one is the natural lipid. This is the overlay between the 4 degree APO and 4 degree capsaicin, the bound structure. There are some local conformation change, but it's not significant. Also, the gate of the TRPV1 are still closed. And then we solve the TRPV1 structure at 25 degrees Celsius with capsaicin. The reason why we choose the 25 degree, 25 degree is the condition, it's very usual condition of the electrophysiology. This is the overlay between the 4 degree capsaicin bound and 25 degree capsaicin bound structure. You can see here, two structures are almost identical. And this is the full focused view of the 4 degree capsaicin bound structure, selectivity filter, and S6 gate. This green one is the 25 degree capsaicin bound structure. Distance is almost identical. Both is the 0. Selectivity filter is the 6.0 angstrom and S6 gate is 6.1 angstrom. Also, we cannot find any conformation change between these two. So that means that this result shown that even 25 degree, the capsaicin cannot activate the TRPV1 in this condition as the same as the heat. Next, we treat both capsaicin and noxious heat at the same time. As you can see here, we obtained the two conformations in one data set. We call this one is the intermediate state and this one the open state based on the pore size. This is the pore focused view of the intermediate state. This is selected filter and S6 gate. And this is the red one is the open state. As you can see here, both gates of this open state are dilated enough for the ion conduction. But the intermediate state, selectivity filter and the S6 gate of the intermediate state is the wider, green one is the 25 degree, wider than 25 degree, but it's not wider enough for ion permeation. Also, this channel opening is a continuum. You can see the clear differences, right? It's going back this way. This pattern is also observed in the conformational chain of the strip domain. Yeah, the closed, intermediate, open. Compared to 25 degree capsaicin structure, both states, the intermediate and open state, show similar global conformational change. I will show you this later. But deeper their local conformation around the pore region, leading to distinct gate states. At this point, we have a question about this intermediate state. Because it is already well known that TRPV1 is easily desensitized by heat. So we could not confirm this intermediate state is an actual intermediate state or desensitized state. So to prove this, we solved another structure with different heat treatment protocol in 48 degrees. These are the model, and the gray one is the cryo-EM density of each condition. You can see here the pore size, both pore size, selectivity filter, and N6 gate. It's clear difference, right? I mentioned earlier, our heat treatment protocol is the 48 degree, the 30-second incubation. However, as you can see here, this is the new model. This time, we treated the heat only 10 seconds, short heat incubation. As you can see this figure, this structure is very similar to 48 degree intermediate state in terms of the pore size, right? However, we do not find any other state, the closed or open state. So we thought the 10-second heat incubation, short heat incubation, was inefficient to open the TRPV1 in this condition. This is very strong evidence the heat-dependent intermediate state exists. Based on this result, we called this the conformation change from the 25 degree to intermediate state as the first transition, and from the second transition is from the intermediate state and open state. And we analyze the conformation change in each state. In first transition, the close to intermediate state, TRPV1 is undergo global conformation change throughout the entire channel. You can see here, the transmembrane domain is moved down, and cytoplasmic domain is moved up toward to the center. This is the close-up view of the cytoplasmic domain. Movement is very clear. It's close to the channel center. And this is the part of this transmembrane domain movement. Overall, the TRPV1 molecule is getting more compact, leading to slight dilation of the selectivity filter and S6 gate. Compared to this first transition, the second transition shows the local conformation change around the pore region. Pore region is consists of the S6 helix and the S6 helix and pore loop and pore helix. Red model is the open state, and the orange model is the intermediate state. You can see here the outer pore of each subunit, the 1, 2, 3, 4, is tilt and rotate on the membrane. This is the close-up view of the single subunit. This one is the pore loop. The pore loop is very important to heat sensing. This pore loop is the move up toward to the outside. This rearrangement leads to the flipping of the methionine 644 side chain and dilation of the selectivity filter. This is the interface between the neighboring subunit. This is the original subunit and neighboring subunit. Interestingly, we observed the rearrangement of the phospholipid bound at the subunit interface. This is the intermediate state. You can see this the Y631 is the flip, and this lipid is going to the interface. And this is might be this. Yeah, we think this lipid conformation change is induced by the rotation flipping of the Y631. Also, S6 gate changed to pi-like helix, not pi helix. Due to the helical chain, S6 gate is more dilated. And then we wanted to analyze the thermodynamic change during the heat transition. Okay, this is the background explanation. This slide. Actually, there is a lot of this temperature sensing model of the thermo-tube channel. Among them, the model proposed by the David Clapham and Chris Miller is accepted as an established theory. According to this model, the heat-dependent thermo-tube channel gating is accompanied by the significant heat capacity change, delta Cp. This delta Cp depends on the change of the enthalpy and entropy. This is the original equation. The delta Cp, we can calculate the experimental delta Cp using this equation. So here, this ASA means the accessible surface area or the solvent-accessible surface area. Okay, this is one of the model. Okay, let's suppose let's imagine this channel is the heat-dependent channel, and this is the closed state, and this is the open state. If it's the one hydrophobic residue, you can see here, it's something like the mucin buried in the closed state and exposed to the dissolvent at the open state. This means that the delta Cp is the positive. Here, this is the kind of concept. So this model suggests there is a considerable delta Cp change when TRPV1 is opening. Actually, this is a very interesting point of view. So we analyze this delta Cp using our structure model. First, we calculate the solvent-accessible surface area, the ASA, per amino acid, and predicted, and we could calculate the predicted delta Cp per residue using this equation for the first transition and second transition. This plot shows each transition, the heat capacity change per single amino acid. This is the first transition, and this is the second transition. This dotted line is the display threshold for the following figure. As you can see this figure, the residue with high delta Cp is more exists in second transition. We map those residues with a high delta Cp in our structure model. This is the first transition, and this is the second transition. Interestingly, although the conformational change is more significant in the first transition, but the heat capacity change of the second transition is much bigger than first transition. This analysis suggests the heat capacity change contributes mainly to second transition. Next, we display the residue with a high delta Cp on the single subunit to determine if a heat sensing domain exists. This is the result, the lead surface. You can see in this figure, the residue with a high delta Cp are pretty evenly distributed throughout the subunit, not specific region. This means that the entire channel, not specific domain, is involved in heat-dependent opening. So we could know there is no specific temperature sensor domain in TRPV1. Last, we tested whether TRPV1 has the same thermodynamic property as the previous proposed model. I think you guys remember this model, right? So we choose one of the residues with a high delta Cp during the transition. It's the N628, which is exposed to the extracellular side. You can see here the closed state is exposed to the extracellular side, to the solvent, and this open state this is buried. So according to this theoretical model, delta Cp would decrease if this residue, the N628, change to less polar residue, such as this dilution, and the channel could be less heat sensitive. So we introduced the isosteric mutation because we wanted to check the only hydrophobic effect, the N2D and N2L. This point mutation, interestingly, you can see here, the point mutation, the N2D mutation, there is the great heat sensitivity, right, compared to this, with the increased polarity, right? So, but while the reducing the polarity, N2L is decreased quite a bit. Actually, there is no the heat sensitivity. When we check this, the Q10 value, Q10 value is below 10. So we know the mutation is almost no heat sensitivity. This point mutation functional effect is correlate well with the direction of the heat capacity change. This is a clear observation. A point mutation can substantially increase or decrease the heat sensitivity of the thermoelectric channel. Okay, this is the conclusion slide. We found the two heat-dependent conformational transition take place in stepwise manner. Also, we found there is no any specific temperature sense domain in TRPV1. And this result is the first experimental proof of the heat capacity change model for the thermoelectric channel. And this further direction is very clear. We want to get the heat open state without ligand. The current structure study is some limitation due to the capsaicin. So we keep studying about the heat open state without ligand. Okay, this is the last slide. I'd like to thank for my lab and at Duke University and our collaborators, especially Professor Seong Lee and the PNCC, the spoke, the Janet Meyer and Lauren and Nancy and Irina, and also our Duke CryoEM Center, the manager, the Relaxi, and yeah, and our collaborator is Mario. So thanks for attention. Yeah, that's what I have.
Thank you for that talk. It was really nice. We do have some questions in Q&A for you. So we have a person wondering what the concentration of capsaicin was used on the structure.
Oh yeah, we used the 50 micromole capsaicin. It's pretty high. Yeah, 50 micromole.
Nice. And then did you try to add PIP2 to see if high temperature or capsaicin alone is capable of opening the channel structure? PIP2 is a cofactor for the channel in a cellular environment.
Actually, this is a very good question, but we didn't add the PIP2. But we tried to get the PIP2 bound state, but we didn't get that state as well. So yeah, it might be worth to try to with the PIP2 and hit together. Yeah, thank you for the suggestion.
I have a question while the audience has time to type up theirs. So I'm curious about when you started plunge freezing at 48 degrees, was information about your parameters for 4 degrees helpful at all? Or were you completely starting from scratch when you were plunge freezing at temperatures like that?
We have some guidance because the electrophysiology is give us some advice. So basically the TRPV1, the threshold is around the 42 degrees. So we start from the 42 degrees, but we kept optimizing.
But I guess in terms of like figuring out your blotting parameters, did you have to start from 0, or was information about what worked at 4 degrees helpful at all?
You mean yeah, that's Okay, say that. Might be. Can you say that again? Yeah.
Yeah. So if you had worked out plunge freezing parameters at 4 degrees, was any of that useful for a starting point for plunge freezing parameters at high temperature, or were you just starting from zero to optimize freezing parameters?
Actually, this is a very good question. We test, we didn't test the high temperature first. So we tested several different temperature. This is room temperature and 37 degrees, something like that. So yeah, we can start this with some. Yeah, maybe the room temperature is the best starting point. So, yeah, 25 degrees.
We have a few more questions. So did you take into account Akron repeats while calculating CP?
Oh yeah, that is this very great question. Actually, we delete quite a bit the anchor lipid domain because we measure the model fidelity about the anchor lipid domain. We're pretty sure this the transmembrane domain, but the anchor lipid domain is the less resolved, especially the high temperature the structure. So yeah, we didn't include. We included it a little bit, but we didn't include the full anchor lipid domain.
And then someone's interested. So did you clone your protein? And if it's a soluble protein, how did you clone it?
Okay, clone the protein. Oh, this is. Oh, sorry, I can't understand this one. Clone.
Is this simply it appears that this is the entire protein including the transmembrane domain. So how did you prepare the sample? Was it cloned? And it looks like full length.
Oh, okay, I see. Yeah. Actually, the TRPV1 is the membrane protein. Yeah. So we initially use the detergent. We extract the membrane from using the detergent, and we reconstituted the nanodisc. Finally, so.
Maybe I can fill in that. Yeah. So this channel is very well established, well studied channel. We didn't clone it, but we use the full length. So there was no modification of the protein.
Okay. Yeah. We had a question in the chat. So the heat capacity model predicts that the channel is both heat and cold sensitive. Is the TRPV1 cold activated as well?
I don't think so. Yeah, the TRPV1 is only activated by the high temperature noxious heat.
Okay. And then here, there are some ideas that partial protein unfolding is important for TRP heat sensing. Are your structures in any way indicative of this mechanism for the reduced resolution in the ARDs?
Oh, yeah. Yeah, actually, this part is the protein part is also we want to know. So we're just starting this collaboration about the protein folding part.
Yeah, actually, can I hop in with a follow-up? I'm wondering, because you don't see any density in that ARD, have you tried doing like focus classification to see, is that reduction in resolution because a rigid domain is moving, or is the entire ARD breathing? I don't know whether that analysis could come out, but have you tried?
Oh, sorry, yeah, I missed that this slide actually. So we already see using this focus classification only the transmembrane domain. So after the focus classification, the ARD is getting worse. Yeah. So yeah.
Right, but have you done the opposite where you do the classification on the ARD?
ARD part. As far as I remember, I tried, but I didn't get any good result. So yeah, maybe I can try it again. Yeah, thanks for a good suggestion.
Yeah. So there's to add to the to fill in the answer that we tried but we just don't see any clear ARD. It's because it's not entire ARD disappear. It's just part of ARD first 4, and so with although we're not for sure, it's not two-state motion or like rigid body motion. It's more like a breathing. I think it's entropy get really enlarged, so increase. So that could answer one of the questions that there should be some. It's possible that the ARD motion could be contributing to heat sensing, but we just at this point it's remain as a question. Yeah.
I have a quick question about the temperature and the humidity. If you have 48 degrees and 80% humidity, the dew point, which is actually probably, as you blocked it, is more relevant than the temperature. The dew point is 43 and 43.5 degrees. Is that still high enough to activate that channel?
Yeah, that part is I've never thought about that. So yeah, I will think about it. Yeah, I'll think about it. Yeah, the dew point.
Well, I think that you're making a good point. I think that there will be idea and to get, but so there was, I think we try to get 42 and 43. We worry about losing heat loss, and we actually we realize there is substantial difference between in vivo in cellular condition versus the in vitro. So we try to get as much as possible the conformation change. So that's why we tried the 10-second and 30-second 48, and we didn't go back down to the 43 or see whether we could actually look at the similar type of conformation. So which is a very good point. So we could look at them.
Yeah, very early on in developing the Vitrobots and the plunge freezing apparatus, they realized that but from the transitions of lipids from lipid vesicles from liquid crystal form to the liquid form, that the important parameter as you blotted and the surface area increased tremendously, the temperature on the grid was most connected with the actual dew point of the dew point temperature in the chamber, and that's one of the reasons. Of course, the other reason, of course, was drying out the evaporation. But the evaporation leads to a lowering of the temperature by evaporation, and so the actual relevant number is the dew point. So so if you calculate it, it's about 43.5 degrees is what the actual temperature on the grid really is at 48 degrees with 80% humidity.
I see. Yeah.
Yeah. Thanks for nice suggestion.
Yeah.
So I think that there's somebody that wants to just confirm the detergent that you used and if it interfered with your results with CryoEM.
We used this digitonin for the membrane extraction, but I think the digitonin itself did not interfere, but the screening result is quite clear. So there is no any intact particle. So yeah, this is not interfere, but that condition is not optimal.
And then the conformational changes observed with the first transitions were already global. Were there subclasses that showed local conformational changes in regions away from the gate? And is it possible that heat triggers some local conformation changes that were not captured?
This one, okay, so let's try. Okay, where is the
I could fill in that it's possible that some local conformation change that could be a trigger that we could have missed, because if it's small conformation change, we cannot really capture because limited resolution and the difficulty of trapping heat-activated conformation. But just this personal opinion is the second transition was quite local. If you look at the global level, and there are very small change between first and second transition, but second transition lead to the channel opening. So I think that's probably the local conformation change we capture, but we cannot be sure. And your point is really valid. And the second question about the functions group, Dohoon, you want to answer that?
Okay. So, yeah. Okay. So what is this? Refer to this chimeric analysis. Show this one. Yeah, this part is it could be the anchor repeated contribute to the heat sensing, but our answer is we're not sure in our study, because the anchor repeat itself is the resolution is not very good enough for the analyze the temperature sensing.
Yeah, I could add a little more to that because of the for those people who didn't know that the background, that the functions group did this chimeric study show that the ankyrin repeat we call the coupling domain, and so junction between ankyrin repeat and the channel domain seems to be very important for heat sensing, and also there are other groups that show that outer pore is very important for heat sensing. If you do our heat capacity analysis, that the solvent accessibility change. Actually, although that heat capacity change group spread out in the protein, that there are localized group that actually focus on an outer pore and also the coupling domain where the functions are predicted. So those seem to be really important. So they probably involved, and also we see that heat capacity change. There's a conformation change we see actually heat dependent conformation change. So they are involved, and so but we think we're not sure whether that act as a heat sensor or entire group that acting more together as a heat sensor. So not just a separate domain, and so that's our idea. Yeah.
I think we have one potential final question. They're wondering which lipids or mixture was used to prepare the nanodiscs and their temperatures, and also if any lipids interfered with the structure for TRPV1.
Yeah, that is also this might be possible. We use the phosphocholine and PC, PE, PG. It's a combination. But we didn't calculate exactly the PM about this lipid mixture. So yeah, it might be possible, but yeah, we're not sure.
But we use a PE, PC, PG, but they have there's a lot of double bond. So their TM is very low, so that is away from the temperature, that the transition temperature is much lower than the temperature experiment that we do. So we don't think that it affects the heat dependent conformation change. This has been questioned a lot in when TRP channel was cloned, and this lipid gel to liquid transition affect the temperature transition or temperature dependent conformation change. It turns out that it's mostly a protein. We need lipid membrane, but the transient temperatures are way below for this temperature dependent conformation change.
All right, I'm not seeing any other questions unless the panel has some other questions they want to ask the speaker.
Yeah, I don't. So I think we can go ahead and wrap up. So thank you so much for that amazing talk. I think you're very brave to set your Leica at 48 degrees. And it's just really cool to see what kind of things you can do with cryo-EM. This is a really exciting system. So thank you for that great talk. And thank you to our audience for joining us today. We wish everyone here well in their cryo-EM journeys. And remember, the national centers are here to support your science. So let us know how we can help. And we hope we see you again next month. Thank you very much.
Thank you.